Thank you, everybody, for joining us today for our Lipid Maps webinar, which is going to be presented by Julie Rumble and Dawn Rundle from Cayman Chemical, entitled Delivery of Nucleic Acids to T Cells Using Lipid Nanoparticles from Chemistry to Expression. My name is Valerie O'Donnell. I'm based at Cardiff in the UK. The webinar is being recorded. It'll be uploaded onto the Lipid Maps website and our YouTube channel in the next 48 hours. Overall, we're usually around 45 minutes with 10 to 15 minutes of the questions, but it may be a little bit longer. See how it goes. I think one of you said to me at the start something about an hour and 15. I think. Let's see. We've got plenty of time anyway, so that's fine. But as we go through, as the presentation goes through, if you guys want to ask questions, can you start posting them during the talk? Don't wait till the end. Put them either in the Q&A or the chat. And that means at the end, what I can do is unmute you and you can ask the questions yourself. So, and we usually take them in the order they're posted. So as you think of a question, put it in the chat then so that you can then be unmuted. Today's webinar has been sponsored by Cayman. We're grateful for their ongoing support. Before Miguel introduces Julie and Dawn, I just want to say a couple of words. I mean, I think this talk came about because of a conversation, at least in part because of a conversation I had with Kirk Maxey back at our business meeting last year. And we were talking about the whole area of lipid nanoparticles and, how this all works, because this is an area that most of us, I think, today at this talk won't be really familiar with. And it's really interesting, a lot of the chemistry and the new lipids and unusual structures, I think, that are part of nanoparticles that although many of them are not natural products, they may well become very important in our lipidomics and metabolomics searches, because as these particles get used more and more for vaccines, for cancer vaccines and anti-infection vaccines, whatever, we are going to be getting exposed to them. So being able to consider how the structures might become more common in mammalian systems through this route is something to consider. So maybe that'll be something we'll chat a little bit about here. So we talked about whether or not over time we need to be adding them to databases so that as they show up in particularly untargeted kind of experiments, we might need to be able to find them. But anyway, this is obviously a really important area. It became very important during COVID pandemic particularly, and now it's really driving a revolution in terms of cancer treatment. I know very little about this. I'm looking forward to learning more. And also, I should say, this is our first industry presentation at a Lipid Maps webinar. And I think this is something we should also do more of. So I'm really excited to hear this. And I'm going to hand over to Miguel, who's also a Cayman, who many of you will know well, I'm sure. And Miguel is going to introduce Julie and Dawn today.
Thank you, Julie. Sorry, thank you, Val. It is like Val said, my name is Miguel Gijon. I'm at Cayman Chemical. I'm a lipid biochemist, and it's a great pleasure for me to introduce my two wonderful colleagues, Julie and Dawn. Dawn Rundle is the manager of research chemistry at Cayman Chemical, where she has been employed since completing her Bachelor of Science in Chemistry at the University of Michigan in 2005. And over the past 5 years, her research has focused on lipid synthesis. And like Val said, these lipids are currently changing our lives, so I'm sure we will hear a lot more about them in the near future. And after Dawn, it will be Julie's turn. Julie Rumble is the Director of Immunology and Cell Biology at Cayman. And after completing her PhD in immunology and a postdoctoral fellowship in neuroimmunology with Dr. Benjamin Siegel, she moved to Cayman to work in assay development. Since then, she has worked to expand the immunology services offerings for Cayman and focus on immunopeptidome analysis, lipid nanoparticle formulation, which she will talk about today, and other immunological analysis. So with that, I will just pass the baton on to Dawn, who will start this presentation, and I hope everybody gets to enjoy it.
Thank you, Miguel and Val, for the introduction. Julie and I are very excited to be here. Thank you, Lipid Maps community, for inviting us to talk today. Our talk today is going to be on the delivery of nucleic acids to T cells using lipid nanoparticles from chemistry to expression. I'd like to give you a brief overview of our talk today. I'll begin by providing a background on lipid nanoparticles, focusing on their lipid components and the therapeutic applications currently in use. After that, my colleague Julie will take over to discuss T-cell transfection using LNPs and explore the exciting potential of LNP-based therapies targeting T-cells. So first off, what is a lipid nanoparticle? Lipid nanoparticles, or LNPs, are composed of a mixture of several types of helper lipids in an ionizable or cationic lipid, carefully designed to facilitate the delivery of nucleic acid cargo into cells. The properties and formations of these particles are determined by the precise defined molar ratios of lipids to each other, as well as to the nucleic acid payload. LNPs are assembled by the rapid mixing of an aqueous nucleic acid solution with an ethanolic lipid solution, allowing the particles to self-assemble. This unique process allows LNPs to effectively encapsulate and protect the cargo, ensuring its efficient delivery into target cells. LNPs have been successfully demonstrated for therapeutic applications such as protein knockdown, siRNA delivery, and antiviral vaccines, or mRNA delivery. Moreover, LNPs hold significant promise for advancing future therapies, including gene replacement and cancer treatments, offering innovative solutions for a wide range of diseases. Lipid nanoparticles are a specific subclass of lipid-based drug delivery systems, and they have distinct characteristics compared with other lipid-based carriers. While all lipid-based delivery systems share the common goal of improving the delivery and efficacy of therapeutic agents, LNPs really stand out for their ability to efficiently deliver nucleic acids. LNPs have a monolayer lipid membrane with a core containing reverse micelle lipid layer surrounding the cargo. Liposomes have a bilayer lipid membrane and an aqueous core. They are widely used to deliver hydrophilic small molecules, but are less effective at encapsulating and delivering nucleic acids. Solid lipid nanoparticles are primarily used to deliver lipophilic small molecules. They have a surfactant shell and a solid lipid core. Due to their solid lipid core, they offer better stability for these drugs, but their rigidity makes them less effective for nucleic acid delivery. And finally, we have nanostructured lipid carriers. They're very similar to solid lipid nanoparticles, differing by containing both solid and liquid lipids in their core. Designing effective lipid nanoparticles involves a number of critical factors to ensure optimal encapsulation, stability, cellular uptake, and targeted delivery of the therapeutic payload. First, one must decide which lipids to use in their ratio. This determines particle size, polydispersity, and efficacy. The ionizable lipid component is specifically important. This lipid plays a key role in encapsulating and delivering the cargo. It also needs to be carefully designed to be metabolized and prevent toxic side effects. Another key factor is the lipid to cargo ratio, which influences encapsulation efficiency. This is often defined as the N to P ratio, which represents the ionizable lipid nitrogen atoms to nucleic acid phosphate ratio, essentially representing the charge balance between the two components. The aqueous buffer used in the mixing process also needs to be thoughtfully considered. It needs to be acidic for the particle formation. However, particle storage and use require a neutral buffer. The particle size can play an important role as well. Smaller particles tend to have longer circulation half-lives and promote better tissue penetration. How it's administered to the patient, most commonly intravenously or intramuscularly, will help determine what cells are transfected. And finally, how the particle is prepared. Hand mixing is cost-effective and widely available, but using a microfluidic device allows for consistency, improved encapsulation, and scalability. Let's take a closer look at the 4 types of lipid components found in an LNP. First, we have the ionizable or cationic lipid. They play a key role in both encapsulating the nucleic acid payload and releasing the payload into the cytoplasm of the cell after transfection. I want to take a moment here to differentiate between ionizable and cationic lipids. A cationic lipid has a permanent positive charge on its head group, while an ionizable lipid is neutral at physiological pH, but becomes positively charged under an acidic pH. The next component is the phospholipid, sometimes referred to as the helper lipid, and mostly present in the particle membrane. They help improve the overall stability of the LNP and modulate membrane fluidity, which aids in the cellular uptake of the particle. They are most commonly a PC or a PE. Next is the sterol lipid, most commonly cholesterol. It is incorporated into the lipid composition to enhance the structural integrity and rigidity of the LNP. It also helps to reduce formation of large aggregates, maintaining particle size. This is all crucial for the stability and controlled release of the carbo. Finally, we have the pegylated lipid, or a polyethylene glycol polymer, usually with a hydrophilic head group. Their purpose is to modify the surface of the LNPs to improve their circulation half-life by reducing particle recognition in the immune system. So now I'm going to take a closer look at ionizable lipids. SM-102 is the ionizable lipid component used in the formulation of Spikevax, the COVID-19 vaccine developed by Moderna, and will be used in a study Julie will present on as well. Its structure consists of hydrophobic alkyl chains, ester linkers, and a hydrophilic amine head group. SM-102 is neutral at physiological pH, but the amine head group is protonated in acidic conditions. This protonation is a defining feature of ionizable lipids, enabling them to interact electrostatically with the negatively charged nucleic acid payload, facilitating its encapsulation. Once inside the cell, the positively charged lipid aids in endosomal escape, releasing the nucleic acid into the cytoplasm, where it can trigger the desired therapeutic effect. Now that I've highlighted some of the key design features that make SM-102 a good ionizable lipid, I want to show how it got there and what's next. DOTAP was one of the first cationic lipids widely explored for use in gene delivery. DOTAP has a positively charged amine head group, making it effective at interacting with negatively charged nucleic acids. However, despite its effectiveness at encapsulating nucleic acids, DOTAP suffers from toxicity due to its strong cationic charge and poor endosomal escape. These issues led researchers to come up with different lipids. Next, I want to highlight D-Lin MC3-DMA, an ionizable lipid that was successfully shown to deliver siRNA and is the ionizable lipid component of Alnylam's drug Onpattro. This cements the shift to ionizable lipids being favored over cationic lipids in LNP formulations. Next, we have SM-102, used in the Moderna COVID-19 vaccine Spikevax. This proved LNP's application to deliver mRNA into cells. As this field continues to grow, researchers are trying to improve upon existing ionizable lipids while also exploring novel structures. One of the next generation lipids from SM-102 is lipid 29. It adds a square amine head group that shows improved interaction with the nucleic acid payload to increase encapsulation. I'm going to talk about another interesting ionizable lipid, C14-4. It's been highlighted in several papers from the Mitchell lab at UPenn as the top ionizable lipid candidate for LNP delivery of mRNA to T cells, and will also be mentioned in Julie's study. The structural features are similar to SM-102, with hydrophobic alkyl chains. This structure has ether linkers instead of the ester linkers of SM-102, but it also has a hydrophilic piperazine core. I won't go into too much detail, but I want to highlight the smart synthetic design of this class of molecules. It's created through a Michael addition reaction between an alkyl epoxide and a polyamine core, which is a one-step process that makes it relatively simple to synthesize a large library of lipid structures. By adjusting the length of the alkyl epoxide chain and using different polyamine cores, the structure can be easily tailored. I want to take a closer look at this class of ionizable lipids. C12200 was one of the first frontrunners of this class. It was looked at very early on as this technology was being fine-tuned. It has been widely studied since. C144 was the next standout candidate of this lipid class due to its success at transfecting T cells. More recently, another lead ionizable structure, C1402, was published that showed having oxygen atoms in the core piece of the molecule can help target where the particles are delivered in the body. The careful design of the ionizable lipid component is a very important factor. Not only does it play key roles with encapsulation and endosomal release, but also potentially with cell type targeting. I want to mention briefly the 3 FDA-approved lipid nanoparticle-based therapies. Onpattro from Alnylam, approved in 2018, which delivers small interfering RNA to treat hereditary transthyretin-mediated amyloidosis, a rare fatal inherited disease with a mutation in the TTR gene. Onpattro is a gene silencing drug that interferes with the production of the abnormal protein caused by this mutation. The other 2 drugs are both antiviral vaccines fast-tracked in response to the COVID-19 pandemic. Comirnaty from BioNTech and Pfizer, and Spikevax from Moderna. There are a lot of similarities with these therapeutics. Firstly, all 3 use the same phospholipid, DSPC, and the same sterile lipid, cholesterol. All 3 deliver an RNA cargo, and they have a little different but similar lipid molar ratios. Where they differ is in the pegylated lipid and the ionizable lipid components. This field is still in its infancy and has grown rapidly in the current decade, partially due to the success and global spotlight of the COVID-19 vaccines. In addition to the 3 current therapies, there are over 40 clinical trials utilizing LNP technologies for a wide variety of applications, including cancer therapies and genetic disorders, along with viral diseases. There have been hundreds of published ionizable lipid structures in the past decade. The naming of these lipids is often confusing and arbitrary. I have seen several published names for the same structure. A colleague of mine recently remarked how every time he sees SM-102, he thinks of this sphingomyelin class of lipids. I think lipid maps would be a great fit to help provide tools for scientists to navigate this field, whether that be nomenclature guides or, as Val mentioned in the introduction, mass spec databases. And with that, I'm going to hand it over to my colleague, Julie, who's going to discuss CAR-Ts as an application for LNPs. Thank you.
Just a little musical chairs. Thanks, Dawn, for that really, really wonderful introduction to lipid nanoparticles and to provide the basis for talking about CAR T cells as an application for these lipid nanoparticles. So I keep saying CAR Ts. What are CAR T cells? CAR stands for chimeric antigen receptor, and this therapeutic modality has thus far been used primarily to target cancer. It is a highly personalized medicine, using the patient's own T cells to kill the cancer using a modified receptor to a cancer surface marker. How does it work? Blood is pulled from the patient, and T cells are isolated. These cells are then transfected with the chimeric antigen receptor. Then they are activated and expanded. Once they have expanded sufficiently, they are reinfused into the patient, where they get to work killing the cancer that they are targeting. This therapy has seen great success in treating and even curing some intractable blood cancers in a subset of patients, and is currently in trials for other types of cancers. Some limitations to this treatment, as it currently stands, include the cost and time to manufacture the modified cells, and some risks associated with lentiviral transfection. In the hopes of improving outcomes for more patients with this revolutionary treatment, there are some potential alternatives. First, there are some potential improvements to the existing therapy, primarily with regards to the lentiviral transfection. Risks of lentiviral transfection include insertional mutagenesis and secondary tumor formation. Alternative methods of T-cell transfection are being investigated, and LNPs represent a very attractive option, partially due to their more transient expression and having been shown to be less cytotoxic than many other transfection methods. In addition, since many components are already included in FDA-approved drugs, the barrier to clinical use may be lower. A future alternative presents a new therapeutic method, namely in vivo transfection of T cells with CAR constructs. If this is truly possible, this therapy would be far less expensive and far more universal, which means greater access. These are exciting possibilities, but the first step is developing the tools and methods required to transfect T cells using LNPs. To work towards this end, we made LNPs using two of the lipids Dawn described earlier. In our LNPs, we encapsulated mRNA encoding EGFP, as this is a perfect reporter for evaluation of expression by flow cytometry. mRNA was diluted in an acidic aqueous buffer. Two separate lipid mixes were made in ethanol. SM-102 was mixed similarly to Moderna's Spikevax, while C14-4 was formulated as previously described in the literature. The amounts of lipid and cargo in each of the mixes were determined based on either a ratio of nitrogen atoms to phosphates in the RNA backbone, as in SM-102, or as a weight ratio of total lipid to total mRNA, as with C14-4. At Cayman, we use microfluidics to formulate LNPs, which means that the acidic aqueous fraction is mixed with the ethanolic fraction at a 3:1 volume ratio by controlled injection into a chip with architecturally defined channels. The mixing geometry, together with the acidity of the aqueous buffer, ensure the self-assembly of the particles in a consistent manner. In the acidic buffer, the ionizable lipid is protonated and captures the mRNA through charge interactions. These methods result in a solution of monodisperse LNPs. To ensure that the particles generated are of high quality, we perform biophysical testing. Dynamic light scattering, or DLS, is a method for measuring the average size and dispersity of the population of LNPs in solution. These two parameters are frequently shown on the same graph as shown here for both of the LNP types. On the left axis and in the bars, you can see the diameter measurement in nanometers. The typical upper range of tolerability for LNPs is around 150 to 200 nanometers, although it depends on the application. These particular LNPs are falling around 70 and 90 nanometers, respectively, which is reasonable for these formulations. PDI, or polydispersity index, which is shown on the right and in the points on the graph, is a measure of the range of sizes of the population. 0.2 to 0.3 is often cited as the upper limit of tolerability, and both of these LNPs fall at or below that value. In addition to the size of the particles, the success of encapsulation is an important parameter. This is tested using a dye that fluoresces in the presence of RNA. With intact particles and successful encapsulation, the fluorescence is low. Addition of detergent releases the cargo and makes it available for binding the dye, which results in a measurement of the total RNA, both inside and outside of the particles. Comparison of the free RNA with intact particles with the total RNA can provide the percent encapsulation, which is shown here on the left axis of the graph and in the bars. Both LNPs had very high encapsulation in the range of 95%. The addition of a standard to this assay can also give us the ability to calculate the total encapsulated RNA concentration, which is shown in the points and using the right axis. This number is used in cell studies to normalize the amount of RNA in each sample. So now that we have particles, we can see how well they transfect T cells. For our first experiment, we used a T cell line called JERKIT, which proliferates in the absence of any exogenous stimulus. We plated the cells and transfected with several doses of LNPs in full serum media. At daily time points, cells were harvested and tested for GFP expression by flow cytometry. I'm going to explain what a flow cytometry plot looks like because many of you may not be familiar with this kind of data. What I'm showing down here at the bottom is a histogram plot with untransfected and GFP negative cells shown using a gray histogram, and GFP positive transfected cells shown in a green histogram. The GFP intensity is on the x-axis, and that allows us to show, determine both the percentage of cells which are expressing GFP, as well as the intensity of the population. As you can see, JERKITs transfected with SM102 at all of the doses tested displayed significant levels of GFP expression, both by percentage of total cells and the mean fluorescence intensity of the population. We continued to assess GFP positivity for 2 more days after SM102 transfection and found that levels peaked at 48 hours, but remained high through at least 72 hours post-transfection. Viability was slightly affected by LNP treatment in a dose-dependent fashion, but the majority of the cells remained viable. We also treated the JERKIT cell line with GFP encoding C14-4 particles and performed the same analysis. By both percentage positivity and mean fluorescence intensity, the GFP expression started high at 24 hours, peaked at 48 hours, and remained high through 72 hours. Viability with C14-4 transfection was largely unaffected, save for a small decrease at the highest dose and longest time point. These data suggest that LNPs can be very good at transfecting T cells, a notoriously difficult to transfect cell type. However, it is important to also test primary cells, as the methods here are a little bit different. A significant proportion of circulating white blood cells, commonly called peripheral blood mononuclear cells, or PBMCs, are T cells. So they are readily available from healthy donor blood. However, these cells are generally quite quiescent and cannot be transfected without activation. In our workflow, after enriching the PBMC population, we depleted monocytes by adherence to plastic and activated the remaining cells using plate-bound anti-CD3 and anti-CD28. Our first experiment aimed to establish the appropriate timing for activation and transfection of these cells. We used the same analysis workflow as with the Jurkat cells, but with an additional time parameter for cell activation. Shown here are cells which were activated with anti-CD3 and anti-CD28 for 3 days prior to transfection with SM-102. At 28 and 48 hours after transfection, cells were harvested for flow cytometric analysis of GFP expression. In this case, while expression was relatively low at 24 hours of transfection, GFP increased dramatically after 48 hours. The kinetics of expression were different when transfection was initiated after longer activation times, which highlights the necessity of testing this time course comprehensively. For further studies, we chose to continue with the 3-day activation and 48-hour transfection workflow. We tested the dose responsiveness of SM-102 particles on T cells using this workflow and found that a broad range of doses resulted in transfection of nearly all of the cells, as well as an increasing level of expression with increasing dose. There was again a small impact of viability of the cells with increasing LNP dose, but the majority of the cells remained viable. We additionally used the same timing model to test C14-4 mediated expression of GFP in T cells. With increasing dose of LNP, we observed increasing numbers of cells expressing GFP, as well as increasing mean fluorescence intensity. Notably, there was effectively no toxicity at any dose of C14-4, as viability was maintained at the same level as untreated cells. So far, we have shown that both SM-102 and C14-4 based LNPs are highly effective at transfecting both primary and immortalized T cells in vitro. Both cell types showed high expression and low toxicity. However, it is worth noting that the typical CAR T generation workflow differs in some key methods from the workflow we initially established. Clinical methods include utilizing purified T cells rather than the mixed population we used, and use bead-bound antibodies rather than plate-bound. So we decided to test changes to our workflow that would more closely mimic clinical CAR T generation. So in order to prepare the T cells, we used Acadium Life Sciences Human T Cell Selection, Activation, and Expansion Kit. The CAR T workflow for this kit starts from human blood leukopaks and adds anti-CD3 and anti-CD28 antibodies to both positively select and activate the cells. These antibodies and the cells they label are captured with microbubbles that float the cells to the surface of the solution. Unwanted cells can then be removed from the bottom, and the cells and the microbubbles are cultured together. As the T cells expand, they settle to the bottom and can be collected for further therapeutic formulation. We incorporated this kit into our existing workflow by starting with peripheral blood mononuclear cells again, labeled with the antibody cocktail. Addition of the microbubbles floated the T cells to the top of the cell suspension, and unwanted cells were removed from the bottom. After several days activation and expansion, LNPs were added, and transfection was analyzed by flow cytometry in the non-bead bound fraction. Cells prepared in this manner proliferated massively over the course of the experiment and were successfully transfected with SM102 particles. We observed the highest expression at 24 hours after transfection, which decreased with time and also with lower doses of LNP. So the goal of the work that we did here was to develop and provide tools and methods that will be useful to researchers who are investigating CAR T cells. The next steps that we would like to look into are to test other LNPs in the purified T cell workflow, such as C14-4, and then also the next generation of lipids like lipid 29 and C1402 that Dawn mentioned earlier. On the immunological side, it will be important to further evaluate T cells in their efficacy post-transfection by looking at things like proliferation capacity and exhaustion markers, as these can impact their efficacy in vivo. Finally, an alternate cargo type may be very important for translating this to the clinic. Plasmid DNA is a much more accessible cargo than mRNA, but it has cytotoxicity issues and different LNP requirements. And with that, I would like to thank the sponsors or the hosts of this meeting, and welcome Dawn back over here, and we will be happy to answer questions.
Great, thank you, Dawn and Julie. That was really interesting. We have a question, and I've got a couple of questions too. Brad German has a question.
Yeah, I just know that you mentioned, like with the microfluidic device, you mentioned that the chip architecture. Are you aware of any studies that have looked into the different types, or have you formulated LNPs with different type of chip architectures? And do you think that's anything that would vary any results based on the architectures?
So I think that chip architecture plays a key role in some aspects of the formulation. We have found that chip architecture can affect the ability to encapsulate certain kinds of LNPs depending on their contents, and different types of chip architecture can make larger or smaller LNPs. The channel size can make a difference. So yeah, I think it's just one more area that you can use to make or test different kinds of LNPs.
Okay. A second question from Suman Alishetty.
Hi, it was a wonderful lecture. So I have a couple of questions. Basically, you have chosen as a golden standard is SM102. Parallel, you are trying C14-4 has a new lipid which has more hydrophilic head. Do you think because of these nitrogens, single nitrogen for the SM102, and because of these multiple nitrogens, so it is more prone to for this T cell kind of activity? That's the first question. And I have seen only one example of C14-4. I didn't see repeatability like one or two different LNPs at the same time. And the third thing is for the R&D scale up is good, but I'm wondering how about if you scale it up in future purpose?
Okay, do you want to start with the first question? I think there were three.
Yeah.
You want to comment on the chemistry side of the nitrogens and...
Yeah, so I think it's interesting the number of nitrogens in the structures. Yeah, and that was one of the things I kind of wanted to highlight with those two examples. And if you'll notice when Julie was talking about formulation, she was using known formulations, whereas with SM102, they specifically formulate the cargo and lipid ratio based on the single amine to the, number of phosphates in the nucleic acid payload. Whereas when she was talking about the C14 formulation, they did it as actually a weight ratio, which was interesting. And I think because of the number of nitrogens, maybe they're not really sure exactly how they're all interacting.
Yeah. Yeah, I think there's a lot unknown still. There are a lot of papers describing a lot of formulations that do a lot of different things, and we as scientists want it to be easy. We want it to be C14-4 works in T cells, and SM-102 works in liver, and ALC works in and it's not that simple. And there are things about C14-4 that may aid in transfecting T cells over some other lipids, but it's not an all or nothing. That is something that we have definitely discovered over the course of these studies, is that you almost always get some transfection, right? It's never no transfection. These LNPs are really good at getting into cells and expressing a cargo.
Because comparing to single nitrogens with multiple nitrogens, the effect of oxidation is also triple, right? Nitrogen is one single nitrogen with N oxide, with multiple of nitrogens, multiple N oxides. The more number of N oxides, the more amount of degradation. The more amount of degradation, the more amount of toxicity. So that's the question to the statement here.
Yeah, I think despite that intuitive thought, C14-4 has really, so far, what we've seen in the literature, it's not been toxic, which is interesting.
Yes,
it's
highly untoxic.
Highly untoxic.
And I mean, we've also found that it is highly stable. I mean, we've had it in our catalog for quite some time, and it's in ethanol, and I don't believe that we see degradation. Some of these other ionizable lipids do see some degradation, but I don't believe that we've seen that with C14-4. So, formally that is a possibility, but just in our experience, I don't know that that's actually happening.
Okay, thanks for that, Suman. Okay, Brendan, you should be able to unmute now. Go ahead.
Thank you, guys. Excellent talk. Really enjoy that. So my question is, and you may have answered a bit of it now, I'm not too sure, but if this has to go in vivo, and depending on what you're targeting, whether it's a tissue or organ, or to remain in circulation and having slow release, after actually delivering the contents, what would be the fate of the lipids that were used to make the nanoparticles?
That is a good question. I mean, some of these lipids are much more biodegradable than others. And so they will be degraded into, at the very least, their components, right? But I do believe that there are probably components that will be found in tissues. And so that's maybe where the lipid maps community can really really contribute to the field, looking at what happens downstream. Mm-hmm. And what of those things that happen downstream, what is important, what is impactful?
Yeah, that's an interesting question. Okay, thank you, Brendan. So I've just got a couple of things to wrap up with. One thing that I kind of wondered about as you were talking is that obviously, small lipid particles like this are all viruses or another form of small lipid particles like this, and they target themselves to tissues using proteins which link to receptors. Is there a way to do that with lipid nanoparticles? Put a protein in and target it somewhere?
Yeah, we're actually doing a lot of that right now. That's a really area of active investigation right now. A lot of people are looking at things like peptides or proteins or antibodies to use those to target LNPs to specific cell types. So the way that I kind of think about it, which is perhaps an oversimplification, is that the ionizable lipid can get you sort of to the right tissue, but then it's going to take a targeting ligand of some sort to actually get you into the right kind of cells. Now, whether that's a peptide or an antibody or a ligand for an existing receptor on those cells, it's going to depend on the system.
Yeah, completely.
So I think the most attractive way of approaching that right now is using click chemistry. And so you can include a one of the click partners as a DSPE PEG with an activated moiety on the end of the PEG, and formulate your LNPs in that way, and then add the appropriate click protein on the other end. We're actively working on that right now. We haven't solved the problem yet, but we're getting there.
No, I'm sure. And I mean, coming back to viruses, another thing that they use as a way to help with entry, apart from proteins, is PS. But PS doesn't seem to be represented in these nanoparticles. Is there reasons for that? I guess it will have been tried, I'm sure, right?
Yes. Yeah, and there are papers that discuss phosphatidylserine as an effective component for specific types of cells entry. So in particular, macrophages are more inclined to take up LNPs that have phosphatidylserine as one of their components. We did test that. Actually, one of our a service client tested that, and I'm not sure it was found to be particularly efficacious. But there's so much variability, right? Like you change one thing, but there's so many things that you can change in any given, because you have at least 4 components, and you can change their identity, their molar ratio. So it's just the design space is like infinite here, right?
Yeah.
Which is exciting and also difficult.
Yeah. Okay. So before we end, we have one last question that was just posted from Madura. Just curious, based on your experience, how is tissue specificity controlled? Could you answer this in the context of a recent study which showed that Pfizer's COVID vaccine LNPs ended up in cardiac cells, indicating off-target effects?
Yeah, I mean, I think that there, as I sort of alluded to earlier, the ionizable lipid can sort of get you to a tissue, but it's not typically all or nothing, really, in any of these cases. I think that these LNPs can get to a lot of places. I mean, if they're carried through the circulation, and if they're carried by cells, even throughout the lymph, or throughout the body, they're going to get to a lot of different places. I think that this sort of cellular targeting that using ligands that we were talking about may be a way to sort of help to avoid some of these off-target effects. Although I think it really remains to be seen exactly how that can happen.
Okay. Well, that was really interesting. I mean, I think we're out of questions and we're coming up to the end now. The diversity of structures is really interesting. All right. Well, all just remains for me and Miguel. Maybe you want to say the last few words and say goodbye then?
No, nothing extra to add, just to thank Julie and Dawn for a very interesting and I think very different type of webinar for us, which I think should be stimulating for all of us. So thank you.
Yeah, thank you very much, and we'll see you all next month. Bye.
Thank you for having us.
Bye.